Hello, everybody. Good afternoon. I also want to thank the organizers for inviting me to this fantastic meeting. And as you have heard already, this is going to be a talk about phages and biofilm and quorum sensing. So it takes up a number of topics that we have heard and are going to hear in this meeting. All of this started very easy, as a PhD side project. We had very little preconceptions and ideas what's going to grow out of it, and I'm going to take you with me on this journey. So we are working with Enterococcus faecalis and Enterococcus faecium. And in the previous talk, you heard about Staphylococcus aureus as a nosocomial pathogen. Enterococcus is sort of similar as Staph aureus in being a very important and complicated nosocomial pathogen involved in a whole number of clinical relevant infections. One of the important things about Enterococcus, similar to Staph aureus and Staph epidermidis, that it is associated with foreign body infections like intravenous catheters, etc. And as you may know, this often also means biofilm. It's a normal commensal of the human gut. We probably all carry Enterococcus in our gut in low numbers. And for a long time, it has been known and it has been thought that Enterococci use quorum sensing. There's a number of two-component systems described, and very early on, LuxS has also been found in Enterococcus. So Enterococcus has LuxS, and that means, or that we thought means that it probably uses autoinducer 2 as a signal molecule. And that we are going to hear more about autoinducer 2 tomorrow, but it's here only a quick introduction. It's a molecule, it's a farnesyl borate diester, and it's formed here by this cycle that's very important in metabolism, involved in methylation. The enzyme that I'm talking about, LuxS, has been used or has been seen as the key enzyme producing the precursors of AI2 in a number of species. Currently, it's more than 70 different bacterial species that harbor this pathway in LuxS genes. So there was a good chance that Enterococcus has also autoinducer 2 as a quorum sensing system. So these systems work, and this is only a quick cartoon, very superficial. So you have a situation where a propeptide is produced, the autoinducer molecule is produced outside the cell, and the neighboring cell takes it up, phosphorylates a transcriptional regulator, then genes are switched on that produce more autoinducer, and the more bacteria you have, the more of these molecules accumulate outside. So what are these for? I mentioned already that these molecules, this signal system is important for biofilms. And biofilms are used by many bacteria in the normal environment, but also in the hospital, because they help bacteria to form complex structures on different substrates. They help to attach to a surface. They help to establish an establishment with the environment. And of course, these thick biofilms protect them against external stress, against the complement system, against white cells, etc. In the biofilm formation in clinical strains, a number of things are involved. They can be adhesion molecules, proteins, in gram-positives. Lipoteichoic acids have been shown to be important for biofilm formation. Extracellular DNA is important to stabilize biofilms, and also other extracellular polymeric substances. And this is a little cartoon that probably you have seen already here, how these biofilms occur. So you have few bacteria that attach to a surface. This green molecule or these yellow molecules mean these are the adhesion molecules that help the bacteria to attach to the surface. The biofilm maturates and then they form larger structures, and you see the gray or the light blue areas are the extracellular substances that are built. And then as the last step, these biofilms dissolve, or it's called they disperse. So the dispersal of biofilms is also important because here bacteria are released, are transferred through the organism and can seed other surfaces. So with the idea in mind that Enterococcus uses autoinducer 2, we started out, we collaborated with Kirsten Jung from Munich, who has worked on autoinducer 2 in Vibrio. And together we did a growth curve, so we grew the bacteria and we measured the autoinducer in the supernatant. And as you can see here, the amount goes up in the first 6 hours up to about 100, between 100 and 120, then eventually falls down again. And here is the mutant that we produced in one of the genes that I showed you before, PFS. These bacteria can't produce any AI2 anymore, and you see here, this is the open circles. They don't produce. The constitution of the bacteria really restored biofilm formation. So this was pretty good and nice, and this is what I just showed you. So this is the circle that I showed you. It's not the LUX-S gene that we inactivated, but the gene before that. This means no autoinducer 2 is produced. We then started to see an add exogenous autoinducer 2 to growing cultures to see what happens. So we have this bacterium. It's an Enterococcus faecalis strain that forms a good biofilm. And you see the biofilm assay is basically the one that you have also heard about in one of the previous talks, where you grow the bacteria in a microtiter blade. The bacteria attach to the plastic. You empty the wells and you stain the plastic, the bacteria that have been attached to the plastic. And as you can see, if you increase the amount of autoinducer 2 that you add to this assay, biofilm increases up to 20 micromolar. But if you use the higher amounts, 50 and 100 micromolar, you see that the biofilm is reduced and actually is also dissolved, because here you have less biofilm than here. And this is also what you can see on this slide. It's an electron micrograph, a scanning electron micrograph. So you see here the thick biofilm that is produced by 20 micromolar exogenous autoinducer and the dissolved biofilm at the higher concentration. So basically going back to what I've showed you before, we have this situation. So you have in the beginning probably very low concentration of autoinducer 2, I've showed you in the graph, attachment of bacteria. And then you have the biofilm maturation, and this is probably what occurs at 20 micromolar autoinducer 2, as I showed you, forming a strong biofilm, and then the dispersal of the biofilm at the higher concentrations. So we wanted to know what happens at these 20 micromolar autoinducer 2 concentration. So what we did, we used bacteria. We added 20 and 100 micromolar of autoinducer, and then we did RNA sequencing to see which genes were upregulated. And we found a locus that's shown here that was strongly upregulated at 20 and downregulated at 100 micromolar of exogenous autoinducer 2. And these genes have been known and have been studied. They are general stress proteins, and there have been several papers describing them to be involved in stress responses, stress-inducible pleiotropic effects on cell morphology, stress sensitivity, and gene expression in Enterococcus faecalis. And although there has been quite a number of papers who have looked at this, the exact mechanisms, how these genes work, has not been elucidated. So we used a mutant in one of these genes, EF80, the first of the two. And what you can see here, this is the mutant. This mutant completely lost the response that I've showed you before. So this is the biofilm formation, as I showed you before. So we don't see this response that the biofilm formation goes up in the mutant. The dispersal of the biofilm still occurs. And we looked also at another function, and here this is a TNF induction assay. So bacteria, if you add them to raw cells, they cause TNF release of the raw cells, and this is what we did in this assay. You see the wild-type bacteria do that to some extent, and here you see that the 20 micromolar AI2, in contrast to here, they downregulate the inflammatory activity, and 100 again upregulates. And again, here you see in the mutant that I showed you, this response was lost. So the hypothesis is now that during the initial states of colonization or infection, the bacteria have to be more resistant on one hand to the host immune system, but on the other hand, they don't want to be as inflammatory. They want to fly under the radar, and later in the process, when cell densities are high and biofilms have maturated, then they want to be more pathogenic. And to look for this, we used an opsonophagocytic assay where we added also the autoinducer to the assay. The assay works, you put together granulocytes, bacteria, antibodies complement, and you let that go for a while and you see how well killing is. And if we did this assay, you can again see here the difference that we saw. In the wild type, we see that opsonophagocytic killing is reduced, so the bacteria at 20 micromolar are more resistant to opsonic killing, and this effect, again, in the mutant in the GLS protein is lost. And finally, this is another assay that we did. This is an adhesion assay where we put bacteria onto eukaryotic cells, counted how many bacteria attached to the cells, and you see here, in this initial assay, the bacteria attached less at 20 micromolar, more at the higher concentration. And again, also here, this effect, this differential effect was gone when we used the mutant in this GLS gene. I would summarize that as I have here. So again, initiation of biofilm formation, some inflammation, some resistance to opsonic killing, some tissue adhesion. All of this increased here, stronger biofilm formation, no inflammation, as I've showed you, resistance to opsonophagocytic killing, and a little tissue adhesion. Now we are going to look at the effects at the higher concentration of autoinducer, 100 micromolar, the dispersal. And what we found when we looked at the RNA-seq data of the addition of 100 micromolar AI2, we found that one prophage of Enterococcus faecalis was highly upregulated. It's called prophage 5. There is altogether 7 prophages known in the strain that we have used. It's a fully sequenced strain. So the idea was really that this induction of the prophage really leads to lysis of the bacteria and release of the phages. And here is what I'm showing you for the effects of the 100 micromolar AI2. So this is the high concentration. And to show that this is mediated through this prophage 5, we got a mutant that Pascal Serrault had produced from this strain where the prophage 5 was completely eliminated. And you can see here adding 100 micromolar of AI2 reduces the biofilm, as I've showed you before, and this differential effect, again, is completely lost in the mutant that doesn't have the prophage 5 anymore. This is the adhesion assay that I've shown you. No, this is the TNF-alpha release assay. And again, you can see at the high concentration, we have an increase in TNF-alpha from the mutant, from the wild type, and in the mutant, in the prophage 5 mutant, this differential effect was also completely gone. Here is resistant to opsonophagocytic killing. Again, you can see at the high concentration, we have a higher resistance to killing. They are less killed, so we have, they are more stable. And again, this differential effect is completely lost in the mutant. And here again, this is adhesion to the eukaryotic cells, Caco cells in this case. And again, you see more adhesion, but the differential effect completely lost in the strain that doesn't contain prophage 5 anymore. On this, I'm going to show you that what I've showed you before works also in different strains. So what we did is, this is the assay that I've showed you before. We used the wild type strain containing the 7 prophages. And from the growth that had these prophages, we purified the phages, we filtered them, and then we used these filtered prophages and infected another Enterococcus strain, in this case, a probiotic Enterococcus faecalis strain that's known to contain no prophages. Well, not completely no, but no mobilizable prophages. So as you see here, when we look, and this is actually what we're doing here, is we look at plaque formation. We use the bacteria, we grow them in culture, we induce them with 100 micromolar autoinducer 2, and then the bacteria that contain phages produce plugs. And this is what you see here. When we do this with the wild type strain, the probiotic strain here, no plugs induced with 100 micromolar autoinducer 2, also no plugs. But the probiotic strain that has been transduced with the prophages that I've shown you here, or not the prophages, but the released phages, you now see that this probiotic strain produces plugs. It produces some without being induced, but it produces quite a lot when it's induced with autoinducer 2. To make sure that what we are seeing is not only plugs, but it's really prophages, we used electron microscopy, and here you can see, again, this is the strains that are induced with 100 micromolar autoinducer 2, and you can see nicely here these little moonlanders. Here are the wild-type strains. This is the strain that contains the prophages. This is the probiotic strain uninduced. This is just another picture showing that really they release phages. The question was, why does that happen? As you probably know, it's always been thought or hypothesized that prophages carry and transmit virulence factors and antibiotic resistances. So what we did in this experiment, we compared the wild-type probiotic strain and the transduced polylysogenic strain in a mouse sepsis model. So where we injected, actually not like this, but into the tailbone, bacteria IV into the mouse, let the mouse go for 3 days, and then cultivated internal organs. And I think this is the kidneys. And you can clearly see this is the wild type. We have more colonies, higher colony count in the kidneys of the transduced polylysogenic probiotic strain. We did the same experiment in a different animal model. This is a rat endocarditis model where you expose the carotid artery here. The mouse, you put a catheter into the left heart. secure it, close the mouth, and then the next day you inject the bacteria through the tail vein. And this forms nice vegetations. It's really a model that looks very much like human endocarditis. And as you can see here, again, we could show that the CFU per milligram of vegetations was much higher, significantly higher in the transduced probiotic strain. So in this slide, we looked at these experiments. So the inoculum is the strain, the probiotic strain transduced with the phage supernatant, as I've shown you. And by PCR, we looked and we saw that that 1, 2, 5, 6, and 7 the prophages 1, 2, 5, 6, and 7 were present. 3 and 4 we didn't see, which really fits nicely also with previous publications who show that they are probably not mobilizable. 2 and 6 are present, constitutively present in the probiotic strain, although they can't be released. And so the ones that remain are 1, 5, and 7. 1 and 7 are known to be transduced only together. They don't transfer by themselves. 5 can transfer by themselves. And here you can see we had a situation where we had all 3 of these, 1, 5, and 7, but we had also several. Here's one that has only prophage 5 together with 2 and 6, but as I said, these are not mobilizable. This is another experiment that we did afterwards. So we used the Symbioflor strain again in a mouse sepsis model, and we just could show that the probiotic strain by itself has this colony counts here. If it has 1 prophage, the prophage 5, which we think is the most important one, it's higher. If it has 1 and 7, it's in about the same range. So the idea that we have of the sequence of events in enterococcal infections, and probably in others as well, is that you have an attachment where these blue dots represent autoinducer 2. So there is very little. Bacteria have to attach, they have to form biofilm, which they do. The biofilm grows and forms these columns at the concentrations that would here be like among 20 micromolar. When the concentration, when the biofilm grows and the colonies become bigger, the concentration increases, and if it's about 100 micromolar, then what happens is that the biofilm is released by releasing these prophages that lyse the bacteria and set free these phages that then also swim around and can attack and can transduce bacteria that are in the vicinity. Which also makes, to me, a lot of sense, because at this stage where you have many bacteria around yourself, it's much more likely that you infect, that the phage infects another bacterium than when the bacteria are more dispersed. So to summarize what I've shown you, I think we've shown that AI2 is tightly regulated and is linked to different stages during enterococcal infections. We think that AI2 is a natural prophage inductor, so release of prophages in enterococcus. Prophage release triggers the spread of virulence genes, as I've shown you with the animal experiments where the phages contributed to virulence. This is where I think it's clinically relevant. Infections of originally apathogenic strains in the gut with specific phages that code for virulence genes results in increased virulence in these transduced strains. And last but not least, I want to introduce you the people who did the work. The major part has been done by Friederike. She was a PhD student at the time. Diana, Andrea, and Dominique helped. We had a very nice collaboration with people from the biology department at LMU, Kirsten Jung. We had help from other people in our lab, especially with the sequencing in real-time PCR, and the nice electron micrographs that I showed have been done by Anthony Hendricks in Utrecht. So thank you for your attention.